Oh, ouch. I think I have a cavity. If this has ever happened to you, don't worry, you're not alone. Over 96.4% of people are affected by caries at least once in their lifetime. The World Health Organization states that untreated dental caries in permanent teeth is the most common health condition according to the Global Burden of Disease 2019. Yet it feels like dental hygiene is not given the priority it deserves.
The field of medicine advances constantly, however, the prevalence of caries has hardly reduced in the last years.
Especially people from poor and disadvantaged communities suffer the most from caries and its effects. If left untreated, caries can cause pain, infections, or abscesses and can end in tooth loss. But what is caries and how is it caused? Our tooth enamel undergoes a constant re and demineralization. Normally, the saliva provides minerals and neutralizes the surface of teeth, contributing to the remineralization, whereas acids cause demineralization. In dental caries, the demineralization predominates through increased amounts of acids leading to tooth decay. Acids, mainly lactate, are synthesized by bacteria embedded in the biofilms on teeth. Streptococcus mutans is the main culprit when it comes to caries. It contributes to biofilm formation by producing exopolysaccharides, shortened to EPS, and produces lactate. Therefore, we decided to focus on biofilm prevention and degradation as a form of caries prophylaxis. For this approach, we use a combination of enzymes, antimicrobial peptides called AMPs, and monolaurin. The specific enzymes, namely dispersin B, mutinase, and dextrinase, decompose EPS, which acts as the structural components of the biofilm matrix, thus breaking apart its adhesion and exposing embedded bacteria. The enzymes have been selected to cleave the EPS produced by S mutans, which are beta-1,3-glucan mutan and dextrin. To prevent the bacteria from regenerating the EPS and forming new biofilms at other places, the AMPs are employed. The AMPs interact with the bacterial cell membrane, making it porous, thus leading to the bursting and death of the cell. Our AMPs should have a specific effect against S mutans, and other cells should not be affected. Monolaurin inhibits the enzyme MER-A, which is responsible for peptoglycan biosynthesis, thus inhibiting cell wall formation. The final goal of our project is a product which can be used regularly in one's dental care routine. Possible forms are toothpaste, mouthwash, chewing gum, or dental stripes depending on the target audience. The latter was brought up to us during an interview and might be the best form of application and was also preferred by the experts as it increases the active time of the usage of the components. We focus on elderly and debilitated persons who struggle with daily dental routines. They may struggle to brush their teeth due to motor and sensory issues and would benefit from the use of our product. You should, however, not forget that caries can affect everyone. People with braces or who had orthodontic treatment struggle to maintain good dental health as well. Our survey on caries showed that many children are affected. That's why we believe that our idea can be beneficial to everyone. After all, healthy teeth play an important role in the quality of life, and everyone deserves access to good dental care without barriers or shame. In order to achieve these goals, we needed to design and clone separate plasmids for each enzyme and peptide. We initially wanted to assemble several composite parts for these plasmids through Gibson assembly. Thus, we had to perform various PCRs to generate the necessary overhangs for the assembly. Later, we also utilized biobrick assembly as well as linkage PCRs. A linkage PCR is a rare method used to link two DNA fragments which have overlapping sequences using PCR. The plasmids were then verified by colony PCR, restriction digest, and Sanger sequencing. Before the transformation of the enzyme plasmids into Piscia pastoris, the plasmids were linearized through a restriction digest with SAL1, therefore enabling the integration of the DNA sequences into the his4 locus in the yeast genome. Methanol can be utilized to induce the expression of enzymes, which should then lead to their secretion. The secreted enzymes can be purified through the his tag with immobilized metal affinity chromatography, or IMAC. The peptide plasmids were transformed in an L-arabinose deficient E. coli strain. The expression of the peptides were induced with L-arabinose. Since one of our peptide sequences is fused to GFP, we measured the fluorescence. Cells were disrupted through sonification, and the total protein concentration was measured by a Bradford assay. After that, a CDS page was performed to verify the expression. The protein should then be purified by IMAC following by enzymatic cleavage. This was necessary in order to gain the wanted peptide sequences and remove the other unwanted elements. Monolaurin can be synthesized through the enzymatic reaction of lauric acid and glycerol catalyzed by Candida antarctica lipase B. The monolaurin synthesis should be validated through an enzymatic assay that determines glycerol concentrations, because we had no access to high pressure liquid chromatography or HPLC. For the destruction of EPS, dispersin B, dextranase, and mutinase were chosen after literature research. For each enzyme construct, PCRs were successfully carried out and Gibson assemblies were performed. However, the cloning of the desired plasmid sequences could not be confirmed by central sequencing. Assembling the fragments with biobrick assemblies and linkage PCRs also did not yield positive results. It was concluded that the cloning strategy needs to be refined. In order to clone the individual peptide fragments into the empty vector PBA-D18, the necessary overhangs had to be generated via PCR. After successful PCR, Gibson assembly with different fragments could be performed. Cloning of the desired vectors was confirmed by colony PCR, restriction digestion analysis, and Sanger sequencing. Subsequently, the respective constructs were to be expressed in the L-arabinose deficient E. coli strain PW25113. Unfortunately, the expression did not yield satisfactory results, which is why the induction strategy must be changed for future experiments. Our approach was an enzymatic reaction to synthesize monolaurin by a condensation reaction of the lauric acid and glycerol. The reaction is catalyzed by lipase B from Candida antarctica. However, the experimental design and the method of analysis were not compatible, because glycerol does not dissolve in N-hexane. Yet the synthesis of monolaurin needs to be conducted under hydrophobic conditions. In order to resolve this problem, a chromatographic method should be performed to verify and quantify monolaurin synthesis. In the human practice part of our project, we had a long way to go from a small scientific idea to a biotechnological product that can help people and cater to their needs. Our first encounter with the public took place during the Tag der Wissenschaft, or Science Day at our university. The participants helped us understand how our project is perceived by the public, and there is indeed a great interest and need for it. We also talked to the scientific group from Professor Dr. Beiler at the Fraunhofer Institute that has already worked on caries-causing biofilms. They not only helped us refine our already planned wet lab protocols, but also gave us new ideas and insights from more experienced people in the field. As we discussed the form of application, the experts pointed out that dental stripes are the best option. In addition to that, we created an online survey on caries and dental health, seeking people's opinions on our synthetic biology project. It showed us that caries in primary teeth is abundant, making children one of our target groups. Since we had many young participants, we also interviewed people with age-related oral health limitations and disabilities such as Alzheimer's disease, arthritis, and Parkinson's disease. We especially asked them about what form of application they would prefer, and they favored mouthwash and dental stripes. All in all, we saw that our idea and new approach to caries would benefit a wider range of people, especially people with limitations who cannot maintain their oral hygiene. Through this experience, we changed the details of our scientific approach and improved upon the needed application of our biotechnological product. Everyone has heard of caries and has a basic understanding of what it is, but most don't know how it's actually formed. As already mentioned, we had an opportunity to educate the public at the Day of Science on caries. But this was not the only approach we took to spread the knowledge in caries and dentism. Through our Instagram posts, we educate the public on caries and interesting facts relating to dental health. To make science more accessible to the people, we also informed them about basic lab tools, machinery, and methods that we used. On several occasions, we traveled in our region to expand our contribution to education. We visited the GASB 7X Designer Conference in Freiburg and presented a poster. It was a great opportunity to get to talk about a project with other synthetic biologists and experience a conference for the first time. We visited different schools and taught students in various grades about the cause of caries, the biofilm, and broke down the essential parts of our project for them. In 7th grade, we showcased how our idea would work on our model Zani, using cutout bacteria, EPS, and enzymes. In the 12th grade, we educated the students beyond iGEM and our project and promoted biology as a field of study. The students showed great interest in our project. For students in the early semesters, we organized an iGEM simulation where they learned how to research a topic of interest, read and present papers to other people, and how to perform such a project. Science encompasses more than individual research. It thrives on collaborative dialogue with fellow scientists, explaining and delving into their research. Therefore, we hosted the Baden-Württemberg meetup at the University of Stuttgart. We invited the iGEM teams of Freiburg, Heidelberg, and Tübingen to acquaint with each other's projects and share experiences within a 20-minute talk. We ended the day with a bowling night and card games. Additionally, we participated in the Junior Jam, the European meetup in Münster, Germany. We're also interested in the public opinion about genetic engineering. In collaboration with iGEM Kaiserslautern, we designed and conducted a survey to probe the general opinion patterns. This survey pointed out that people are skeptical about genetic engineering, and therefore more work had to be done on public awareness. If one wants to develop a prophylactic against human pathogens, the question inevitably arises as to how one can carry out a safe proof of concept. In our case, the most valid proof of concept would be testing our components with our main target as mutans. These experiments would be conducted under Biosafety Level 2 or BCL2 because of the classification of S mutans. We contacted the group of Professor Dr. Susanne Beiler from the Fraunhofer IGB in Stuttgart, which are experienced in working with S mutans and have a BCL2 laboratory in their facility. We considered working with the BCL2 laboratory ourselves, but after some issues we had to change this plan and aimed for the experiments to be conducted by the employees of the Fraunhofer IGB. Moreover, we discussed the usage of certain Streptococcus salivarius strains as an alternative organism to S mutans due to the similarity. The strains we planned on using are classified as BCL1 organisms. We also considered performing a cell-free proof of concept experience because of some problems we encountered. The required components of EPS should be utilized instead of a whole biofilm to test the activity of our enzymes. Furthermore, we employed the yeast Piscia pastoris in our project, which can potentially form spores under nitrogen-limiting conditions. We prevented this from happening through the usage of media and agar that was not limited in nitrogen, like YPD medium, YPD agar, and minimal dextrose agar. Additionally, we paid attention to always contain the organism in closed environments, like clean benches and containers. To enhance the results, we are planning to refine our enzyme cloning strategy. We envision engaging with experts and exploring new techniques such as the Golden Gate cloning. Regarding the monolaurin synthesis, the experiment could be repeated when access to an HPLC or gas chromatography device is provided, which allows for its detection. Regarding the poor peptide expression, we assume that the most plausible cause to be a problem with the induction of the expression. Therefore, we want to perform an induction assay. For our proof of concept, we had planned to perform crystal violet staining, proliferation assays of S mutans, cytotoxicity assays for mammalian and non-biofilm forming bacterial cells, as well as determining the minimal inhibitory concentration among other experiments. For that, we consider collaborating with the Fraunhofer Institute. It is also of interest to conduct studies to evaluate whether our agents have any impact on the tooth enamel. Upon the successful completion of these phases, we would then initiate a preliminary compatibility studies followed by clinical trials. Should our product prove itself effective and safe, we would then move forward with a scale-up in a larger production setting and endeavor to bring it to the market. We could envision collaborating with dental professionals who could utilize our product for gentle dental caries prophylaxis. Lastly, we want to thank our professors, advisors, and sponsors for their support in making this project possible. Special thanks to Professor Susanne Beiler and her team from the Fraunhofer IGB and to the University of Stuttgart.